Hello, my name is Adekemi Taylor, and I'm the Senior Director of Integrated Drug Development at Certara. I will be presenting the fifth case study on model interconnectedness, how modeling was used to guide development of a gonadotropin-releasing hormone receptor for the treatment of endometriosis. The case study is based on this paper by Riggs et al. I will provide a brief overview of the analyses. For more details, please refer to the publication. Endometriosis is a gynecological condition caused by proliferation of endometrial-like tissue outside the endometrial or uterine cavity. There are a variety of clinical manifestations of endometriosis, with severe menstrual cramps often being the major presenting symptom. Medical treatments for endometriosis involve suppression of female hormones. In particular, suppression of estrogen stops proliferation of the tissue and can reduce the endometrial-like tissue outside the uterine cavity. Gonadotropin-releasing hormone, hereafter referred to as GnRH, releases hormones responsible for the synthesis and release of female hormones, including estrogen. Therefore, GnRH modulation affects estrogen production. Now, treatments that reduce estrogen can also lead to adverse effects like those experienced during menopause, such as loss of bone mineral density. Therefore, it is important to identify a range of estrogen suppression that balances reduction in endometriosis symptoms as measured by the Endometriosis Symptom Severity Score, ESSS, in this case study, with minimal loss of bone mineral density. Lower ESS scores indicate less symptom severity. The case study presented here is about model-informed development of naferolin, an agonist of GnRH developed for the treatment of endometriosis. The purpose of the work was ultimately to identify an estrogen suppression target for endometriosis treatment. ESSS data obtained from 3 clinical studies of naferolin and published data on bone mineral density were used in the analysis. Specifically, the analysis aimed to answer the following questions. What biomarkers, for example, bone markers, estrogen, and so on, provide reliable long-term predictions of bone mineral density? What are the expected profiles of these biomarkers over time? And can a target range for the selected biomarker be identified? This analysis consisted of 2 models. The first involved efficacy modeling using a logistic regression model to describe the relationship between the estrogen estradiol and the endometriosis symptoms severity score, ESSS. The second was a multi-scale quantitative systems pharmacology, or QSP model, describing the relationship between estradiol, bone markers, and bone mineral density. The integrated results of both models were then used to guide treatment duration, biomarker selection, and target response aspects of the study design. A logistic regression model was used to quantify the relationship between estradiol concentrations and the probability of experiencing different ESSS scores using data from 3 clinical studies of naferolin. This model predicted the probability of a given ESS score or score range as a function of estradiol concentration. The figure shown here displays the fraction of patients experiencing each ESSS score at different ranges of estradiol concentration. Observations are displayed as black circles. The median predictions from the model are displayed as solid lines, and the 95% credible interval of the model predictions, which can be viewed as a measure of uncertainty, are shown by the dashed lines. As we can see, the logistic regression model provided a good description of efficacy across estradiol levels. The model predictions followed the general trends in the data. For example, the probability of experiencing the least severe ESSS score of 0 decreased with increasing estradiol concentrations, and the probability of experiencing the most severe scores of 4 or higher increased with increasing estradiol concentrations. Next, we take a look at the multi-scale quantitative systems pharmacology model that was used to describe the estrogen-related effects on bone markers and bone mineral density. Literature data were used to develop this model. Without going into detail, you can see that the QSP model was quite complex, as evidenced by the ordinary differential equations presented on the left. In fact, these constitute only a subset of the 29 ordinary differential equations that comprise the model. As we can see from the plots on the right, the model provided a good fit to the published data. We see that the model and the predictions of the model are shown by the lines on the plots. The model was able to describe changes over time in lumbar spine bone mineral density and also in the bone markers serum C-telopeptide and bone-specific alkaline phosphatase at different levels of estradiol suppression, which are indicated by the percentages in the parentheses. The plot on the left shows that the model was able to capture the increased loss of bone mineral density at higher estrogen suppression levels. We also see here that the QSP model provided predictions of the relationship between estradiol concentrations and lumbar spine bone mineral density that were consistent with data from various GnRH agonist and antagonist drugs, which are represented by the different colored symbols. To recap, the logistic regression model of ESSS scores served as the model for efficacy, and the QSP model served as the model for safety. Although the two models were not directly connected, with no information exchanged directly between them, the models still work together to give an integrated view of the safety and efficacy profiles as a function of estradiol concentration. We see how efficacy decreases with increasing estradiol levels. The probability of the least severe ESSS score of 0 decreases, while the probability of the worst scores of 4 or more increases. On the other hand, we see how bone mineral density increases with increasing estradiol concentrations. This plot allows the selection of an estradiol target range that is deemed to have an acceptable benefit-risk profile. The selected range of estradiol was 20 to 40 picograms per milliliter, which was deemed to provide an efficacious endometrial pain response while minimizing loss of bone mineral density. I hope I have been able to demonstrate through this case study that different model types using different sources of data can be used to answer different questions in a drug development program. In this case, we had more traditional logistic regression modeling used to model neferalin efficacy, although it was a little different in that estradiol concentrations rather than neferalin exposure drove the effect in the model. The complex QSP model was used to describe the effect of estrogen on calcium homeostasis and bone remodeling, and enabled an assessment of the safety profile as a function of estradiol level. These two models allowed a better assessment of the neferalin benefit-risk profile. This brings us to the end of the presentation. Thank you.